My title is Response Analysis of Odor Sensor based upon insect olfactory receptors using image processing method. We are electrical engineer, and this is a collaboration with biologists. As these two people are biologists actually. And first, this is the background. And so, although many papers related to electric noise have been already published, its sensitivity, selectivity, and robustness against disturbance are still insufficient. And so we are looking for a breakthrough. And so a breakthrough to solve this problem is a biomimetic approach to use OR. OR means olfactory receptor. We adopted an insect cell-based sensor since its signal pathway is very simple and variety of olfactory receptors are easily utilized. Although fluorescence image of biological cells is useful to detect odorant, the effective method to extract information from image is required. So the purpose of this study is that the development of image processing method to raise sensitivity when we use bioelectronic noise. And so this is a sensing principle of biological cell expressing olfactory receptors. This is a cell, and this is a cell name is an SF21 cell. And on the surface of SF21 cells, this is the olfactory receptors. And odorant molecule comes here and binds that. This is ligand-gated ion channel. And so ion channel open, and calcium ion, that one flows into the cell. Here is GCAMP3, that one is a calcium indicator protein. So that one the calcium bind this one, and fluorescence intensity increase. Excitation light here is 488 nanometers blue light, and fluorescence light here is green light, and increase in intensity of fluorescence light. This is around 515 nanometer and 520 nanometers around these regions. And so this is an experimental setup of fluorescence measurement. So we developed our own system here. And this is a laser diode. So this one illuminate 488 nanometers. And so this one is the setup of the laser diode. Laser diode actually here. And so this is the circuit. And that one goes to here. And this one is the dichronic mirror. And that one goes to, light goes this way through this one is objective lens. Here is a sensing chamber with cells. And so this syringe means that some odorant stimulus is injected using this syringe. And after that, fluorescence occurs, so that one goes this way. And so this one is captured by cooled CMOS camera through this one, long pass filters. And so captured image is transferred to personal computers here. And so size is around, height is 470 millimeter, width is 220 millimeters. And so this one is different from fluorescence microscope and laser diode. We use that, and the laser diode enables simple and compact measurement system of fluorescence. And so this one is a grayscale image of SF21 cell expressing OR56A. OR56A is the name of the OR we use, and this one is specific response to the geosmin. And so first, this one the typical image of this system. And so this one, after you extracting the cell region, this one is image after image processing. This is the image after image processing. And so although these two images seems quite similar, actually they have differences. Here, and this one area outside cell. This one is a cell. Area outside cell has some brightness of certain level, even if it seems dark. However, this is not zero. And so this one is that so area outside cell had 0 level. I mean that so this one, so we do the image processing and do that. So this slide explains that so how image processing is why is image processing necessary. And this one is sum of the brightness, and so this one, the original image is here. This one is after extracting cell image, and totally about 70% of here, that one is suppressed, and sum of bright intensity in region without cells was almost 70%, and background noise in region without cell influence signal to noise ratio. And thus, region with intensity change should be extracted. And so this one is the procedure of the image processing. First, we obtain the reference image. Reference image just before sample injection, and image with the largest intensity after injection are prepared. So we obtain 2 images. And so this one's difference image between reference image and the largest intensity image. So that one is obtained. And so the third step is a threshold value is determined using histogram of light intensity change, and total number of disjoint categories, we call that one bins. So that one is 100 in this case, and the threshold is said to be the central value of the smallest bin. And fourth stage, light intensity below threshold is said to be 0. And finally, we use a median filter, which picks up the median value is given region, and is applied to remove noise caused by the difference operation. So this one, we will use a difference image, and some noise occurs due to the difference operation, and the median filter suppresses such a kind of noise. And this one is the optimization of number of bins, and these were original image. So this one is a cell, and so this one is before injection, and this one is after injection. This one is grayscale image, and here so number of bins is 40, and this one is before sample injection, this one after sample injection, but several regions were not extracted here, so threshold is too high. And this one number of bins 100 at that time. So compared with original image, so appropriate image was obtained. So we said the number of bins 100. And so this one means that so cell image of SF21 expressing OR56A. As I told you, so OR56A is specific to the geosmin. Geosmin is a kind of mold smell. And so sample here, and so this is the original image, and this is the image after processing. And A, B, C, so 3 figures. So this one is before sample injection, this one is just after sample injection, and this one is 120 seconds after sample injection. And so we obtain grayscale image here, and so this one is original image here. However, so we pick up the region with intensity change here, and so this one after sample injection, this region picked up here, and this one is a little bit later than the sample injection, and so these regions are picked up. So only area with brightness change was adequately extracted. And this one is the temporal profile of brightness change due to sample injection. The sample is again so geosmin, and so this lateral axis is the time, and the vertical axis is the relative light intensity. And this point here, brightness is normalized by that before sample injections. And blue line means that without image processing, and red one means that with image processing. And you can see that so when we do the image processing, we have high sensor response, and image processing enhances signal originating from odorant stimulus. And so this one is a relationship between sample concentration and relative intensity of light. Lateral axis is the concentration of geosmin, but vertical axis is the peak of the relative light intensities. And so blue plot means that without image processing, and red plot means with image processing. And so when you see this graph, without image processing, around 1 to millimolar, so this one is almost the detection limit. However, that's when we do the image processing, around 1 order of magnitude, so detection limit can be decreased. So detection limit becomes lower after image processing. So this is the conclusion. Fluorescence measurement system using laser diode was established. And image processing algorithm using difference operation, selection medium filter was proposed. And proposed algorithm is useful to extract cell region with brightness change. And proposed algorithm can improve detection limit of odorant. And future work is that image processing will be applied after automatic measurement system. Now we are developing the automatic measurement system, and so we combine these two, and so obtain much data so using this kind of image processing and automatic measurement system. And this is acknowledgement, and so this work was partially supported by Japan Society of Promotion of Science. Thank you very much for kind attention.